everyone. I hope everybody's doing super great and awesome. Right now, I'm back with another session and we need to complete this chapter ASAP. All right. So, in the previous session, we did learn about a lot of things, including the nucleus, endoplasmic reticulum, ribosomes, and your Golgi apparatus, which was given by the Camillo Golgi, discovered by the Camillo Golgi, right? And today we'll be continuing with this chapter and covering these topics, the vacuoles, lysosomes, mitochondria, cell wall, and plastids. Now listen to me on this one. The most asked questions are from the mitochondria and the plastids, okay? Reason being, both of these are semi-autonomous organelles, okay? Semi-autonomous organelles. What do you mean by semi-autonomous organelles? Semi means partial, okay? And autonomous means controlling. So basically, both of these organelles can partially control their own functions, and half of their functions are controlled by the nucleus. So that is why most of these questions are asked from these organelles. Apart from that, obviously, lysosomes are important, right? That does not mean you'll ignore all the other organelles. So we'll be learning about all of them. Now, moving forward, we'll be learning about the vacuoles first of all. If you remember correctly, I did tell you about the vacuoles when we were discussing about the several differences between the animal and the plant cell. Vacuoles basically are the organelles which are present in the center in the plant cell, and they require a huge space of the cell, right? Because they do the storage part of the plant cell, be it the water, be it food, be it different kind of minerals or salts, or be it different kind of different secondary metabolites, or let's say secondary products such as the gums and raisins, right? Whereas in the animal cells, the vacuoles are not present as much, or if they are present, they are already present in the smaller amount or the smaller sizes, right? So let's just go through this first of all, and we'll go by the keywords as you know by now, since I've been teaching you for quite a while. See, vacuole basically is a membrane bound organelle. When I say membrane bound organelle, that means it is covered by a membrane, and that membrane in the plant cell is called as the tonoplast. What is it called? Tonoplast. I already did tell you about this in the previous class. Okay. Now see, vacuoles are also the kind of vesicles. Why vesicles? Because these are also bubbles, right? They are doing the same function as vesicles. They're also storing the things. They're just not bursting like vesicles, okay? Now see, these are closed sacs. That means these are closed compartments, and their membranes could be made up of multiple molecules, okay? Multiple molecules, and inside of that, they can have so many things. They could have the secondary products, they could have the water, they could have the food, they could have the proteins, they could have the enzymes, they could have the salts, minerals, completely dependent upon where these are present, what kind of cell have them, okay? And then there is no set shape or size, because if the let's say content of the vacuole is more, the vacuole will be larger. If the content of the vacuole is less, the vacuole size will be smaller. Similarly, if there are more contents which need to be stored, the vacuoles will be more in number, okay? And cell can change them as it wants, completely dependent upon the type of the contents which need to be stored, okay? And they are present mostly in the eukaryotic cells, and they do multiple things, okay? Like multiple things. We'll be discussing about their functions too, right? So moving on to the vacuole's functions, first of all, these help in the storage of multiple compounds. First things first, which we have already discussed so far, right? Apart from that, these also store multiple ions. Now when I say ions, that means these particular ions, they could be calcium, sorry, chloride, they could be potassium, right? These two are very important for the sake of proper functioning of cell. Why? Because these help in several important metabolic reactions. Remember what metabolic reactions are? Now I'm going to count to 5. You answer me, what is the metabolic reaction? Absolutely correct if you have answered the question right. Metabolic reactions are the reactions which basically are occurring in any living body, be it the human beings, be it the plant cells, animals, microorganisms, etc., etc., right? Apart from that, they have the hydrolytic enzymes that basically help in the digestion of stored macromolecules. What are the macromolecules? Macromolecules are called, like, they are something like the carbohydrates, okay, proteins, basically the molecules which help, which basically are the bigger in size first of all, and then help, and they are big help in the survival of the human being or the multiple organisms, that is. Now, what is hydrolytic enzyme? Hydrolytic enzymes are some things or the proteins which basically help in the breaking down of material. Hydrolytic means breaking down of material. So enzymes are mostly proteins. They are made up of proteins. Okay. So these hydrolytic enzymes are the proteins which help in breaking down of these macromolecules into the smaller particles, which could be easily used by the body, which could be easily assimilated by the body, right? That being said, they also store the metabolic waste of the plant cell. Remember when I told you that these are used for the storage of multiple things, including the secondary metabolic waste, right? Such as the resins, gums, and rubber, which is not actually useful for the plant. Next thing is they could also store the pigments such as the anthocyanins responsible for the color of the flowers. Now listen to me very carefully. Apart from the chloroplast, which has the chlorophyll, there are multiple types of pigments which are present in the plant. There are xanthins, there are anthocyanins, right? There are carotid pigments. All those pigments, they provide color to the flower, they provide color to the fruit, they help in capturing of the different wavelength of the sunlight, right? In health, and they hence help the plant in making the food, right? Now, that being said, in the animal cells, vacuoles, they often associated with the maintenance of water. Okay. Now, this maintenance of water can also be called as the osmoregulation. Osmoregulation is the ability to maintain the balance of water and salt in a body, okay, or a cell, right? If we do not maintain the proper cell water or salt balance, it may lead to the endosmosis or exosmosis. What is endosmosis? When a lot of water enters into the cell through the plasma membrane. And exosmosis is when all the water content of the cell just rushes out of the cell through plasma membrane, right? Both of those can lead to the death in the animal cells because we do not have any kind of cell wall, right? That being said, these work in the osmoregulation, which I have already explained to you, maintaining the osmotic pressure of the cell. Osmotic pressure, as I already explained to you people, the endosmosis and the exosmosis. I'm going to write that down for you in order to avoid any confusion. Endosmosis, exosmosis. Okay. Endosmosis is when cell takes up a lot of water. Exosmosis when cell water contents rush out of the cell, right? That being said, that is all about the vacuoles that you need to know. It's pretty easy. And next we have the lysosomes. Now see, lysosomes are the most interesting organelles. Why? Because these are also called as the suicidal bags. I wonder why are they called as the suicidal bags? No, it's pretty interesting and intriguing. Well, you know what? They're called as suicidal bags because they have a lot of hydrolytic enzymes. What are hydrolytic enzymes? I just did tell you that hydrolytic enzymes are the ones which basically help in breaking down of the complex molecules into the simpler molecules which could be used by the body, right? But the hydrolytic enzymes which the lysosomes have, they can digest the cell itself and hence causing the death of the cell, right? We're going to go with the keywords: contains digestive enzymes, or the hydrolytic enzymes, hydrolytic and the digestive, one and the same thing. Okay. These are involved with various cell processes. What kind of cell processes? I'll tell you. This is like pretty interesting. So whenever we are baby and when we are present in the mother's womb, we do not have fingers separately. Okay. We do not have fingers separately. Rather, our fingers are formed when the cells in between the fingers are digested, or these are destroyed with the help of the lysosomes, and then we can separately have the fingers, right? They also break down the worn out cell parts. Let's say one Golgi apparatus of the cell is not working properly and it is creating problems. So what will cell do? Cell just basically release the lysosomes onto the Golgi apparatus, which is not working properly, and that will be digested. Right? So lysosomes also help in the maintenance and the reviving of the cells. And next, they could also be used for destroying the various viruses and bacteria. Now, how does this happen? This basically happens with a factor whenever the cell kind of senses the entry of the virus or a bacteria inside itself, it just starts on destroying those. How? It releases the lysosomes. Okay. And the bacteria or the virus come from the outer side, and they both fuse, and when they fuse, the virus or the bacteria are trapped inside the lysosome in our cycle, where they are digested and killed, right? But if the cell senses that it has been infected properly, it will start and trigger the suicidal attempt. Okay, it will just start triggering all the lysosomes to burst open and digest itself, right? So that is why they're also called as the suicidal bags. Apart from that, do you know how lysosomes are formed? You'll be pretty amazed. Lysosomes are formed with the help of Golgi bodies. I know, right? Golgi bodies, they are responsible for forming the lysosomes, and lysosomes are not as simple as they look to you. Okay, lysosomes basically have four different types, which will be starting. Now, it is not as necessary for you to memorize all four, but it is pretty interesting and easy as well. Okay, so we have the primary, secondary, residual, and autophagic lysosomes. So we're going to do the types and then we're going to do the functions, although I've pretty much explained all the functions to you. Okay, and I hope you're noting everything down. If not, watch the recording again. Anyway, back to the types. We have the primary lysosome. We have the secondary lysosome. We have the residual bodies, and we have the autophagic vacuoles. Okay. Now moving forward to the primary lysosome. Primary lysosome is basically the newly formed lysosome from the Golgi apparatus from the trans phase. Remember when I told you the Golgi apparatus has like two phases, cis phase, which is the receiving phase, and trans phase, which is the dispatching phase. Okay, and from the trans phase itself, the lysosomes are formed. So the primary lysosome is the newly synthesized. It is newly synthesized. Okay, and what does it have? It basically has the enzymes, okay, which are synthesized by the rough endoplasmic reticulum. Why rough endoplasmic reticulum? Because it is responsible for synthesizing the proteins, and most of the enzymes are nothing but the proteins. Okay, so it'll be having newly synthesized, newly formed enzymes. Okay, now remember this, that these enzymes are inactive. Okay, they are inactive in the primary lysosome. Why are they inactive? Because the primary lysosome is just formed and it does not have any target yet. Next is the secondary lysosomes. Secondary lysosomes are also called as the digestive vacuoles. Okay, aka digestive vacuoles. Now, why are they called as the digestive vacuoles? Listen, I'll draw a diagram for you right here and you'll understand. Suppose this is a cell, Okay, and this is a virus which is standing outside the cell and it is trying to enter it. Now when it enters, what will happen? It will be entering into it in the hair, in like inside it, in the form of a vesicle. Okay, because it will be entering through the plasma membrane and it will just be let's say captured. Okay, it will be captured inside that particular. So let's say if this is a virus, look at my video, let's say this is the virus and this is the plasma membrane which is trying to capture. So when it tries to enter it, the plasma membrane will be surrounding it. Okay, and after it has surrounded it, it will just be moving inside it in the form of a vesicle. Okay, it will be moving inside the cell in the form of the vesicle, pinching off from the entire cell membrane. That part of the cell membrane, it'll be pinched off with the virus inside it. Okay, and this pinched off vesicle will be called as the phagosome. Okay, phagosome, it will be called as the phagosome. Right, now what will happen, we already have the primary lysosome inside the cell. Now, when the phagosome and the primary lysosome, they both combine, that is called as the secondary lysosome, and it has the activated enzymes because the target is already present inside it. Okay, and then we'll have the phagosome plus primary lysosome. Isn't it easy? And also activated enzymes. Activated enzymes, right? Next we have is the residual body. Now listen to this. Residual body is like super easy to learn because what is residual body? As the name suggests, all the digestion which will happen inside the secondary vacuole, all the undigested material which will be present will just remain inside it. Okay, and that particular vacuole with the inactive enzymes, again the enzymes will be inactivated as the target will be finished. So inactivated enzymes and the undigested material will be left with the secondary vacuole or the secondary lysosome, which will be called as the residual body. So these secondary lysosomes left with undigested material and inactivated enzyme are called as the residual body. That means that let's move forward to the most important type of the lysosome. We have the auto self phagic eating or destroying. Okay, self destroying or eating vacuoles. These are the ones which are called as the suicidal bags. Now what will happen if the cell senses something's wrong, it's with itself, it will trigger the autophagic vacuoles, and these autophagic vacuoles will actually be bursting into the cell, and they will engulf all the cell organelles. They'll kind of let's say phagocyte or engulf all the cell organelles and start breaking down their proteins and all the components, and hence will cause the death of the entire cell. That is what the autophagic vacuoles are. Now we know the functions which we discussed in the first place. Now we know the types and we know how exactly they are helping us. Okay. Next is the mitochondria. Mitochondria is the semi-autonomous organelle, which I did tell you about in the beginning of this particular session. So mitochondria are the membrane-bound organelles, but one thing that you need to mention right here is that these are double membrane-bound organelle. Okay, how? I'll tell you, just like the nucleus. Now these are double membrane-bound organelles, and these are the powerhouse of cell. Powerhouse of cell. Most importantly, they are called as the powerhouse of the cell because they generate ATP, which is the adenosine triphosphate. Now you need to memorize this adenosine triphosphate because you're going to have to learn this for the longer run. Okay. Now see, They generate most of the ATP, which helps in all the cells' biochemical reactions or the metabolic reactions. On top of that, since the mitochondria are the ones which basically are the semi-autonomous organelles, that means they partially control their own functions. How do they do that? They do that because they have a bacteria like DNA, or let's say they have a primitive old or ancient DNA or small fragment of DNA in themselves. Okay, and mitochondria in a person are mostly inherited from the mother, and if that happens, like usually that happens, so when that happens, studying the mitochondria, we can actually go ahead and study the lineage or the line of your mother's family, which is quite interesting if you ask me, right? Anyway, so these are the semi-autonomous organelle. The most interesting thing that we are going to do today is going to study the mitochondrial structure, okay? And mitochondrial structure is the easiest one to learn. Apart from that, people can ask you the structure of mitochondria to be drawn during the exam, or you can be asked about the different kind of labelings of the mitochondria in the multiple Olympiad exams. Okay, now that being said, just move forward to the mitochondria. Okay, let's study a bit about what kind of history the mitochondria has. I'll just give you the name of the person who coined the term mitochondria. So it was Benda in 1900 who gave the term mitochondria. Why the term mitochondria was given? Because mitos means thread and chondrion means granules, a thread of granules. Okay, why do granules? Because mitochondria has a lot of granules which help in the production of the energy which is used in the working of the entire cell, right? All right, moving forward with the mitochondria structure. Okay, I hope you can see this structure very, very clearly and properly. I'll be mentioning everything alongside explaining this particular structure. So it is a double membrane bound organelle. I just did tell you about this because it has the outer membrane and it has the inner membrane. On top of that, it has the intermembrane space. Now, this intermembranal space is very, very important. It's very crucial. Why is it important? Because this helps in production of ATP or the production of energy. Okay, understand this point that ATP is the universal unit of energy. It is produced in all the living beings on this planet, be it amoeba, be it human being, be it a plant, be it a bear, be it a lion, etc., etc. Okay, and on top of that, inner membrane is very, very important. Why? Because it has oxysomes or peroxisomes peroxisomes or ATP synthase attached to itself. Okay. Now, what are these different names that I've just taken? What are these? These are the actual names of the part of the organelle, this organelle, which help in the production of ATP or they produce ATP. Okay, so these are like really, really important. And then we have the crista. Now, crista is nothing important but the folding of the inner membrane into the, let's say, folding of the inner membrane in order to accommodate it inside the outer membrane. And why the folding of inner membrane is very important? First, to increase the surface area in order to have multiple peroxisomes attached to it. Secondly, to accommodate itself inside the outer membrane, right? Then we have the phospholipid granules. These phospholipid granules, they basically help in producing the membranes, vesicles, and biogenesis of mitochondrial membranes. Since you see that mitochondria has about two membranes, inner and outer. And then we have the circular DNA. I did tell you about the circular primitive type of DNA which is present inside the mitochondria. Primitive means old or the ancient. Okay. Now, this particular DNA is circular, unlike the modern DNA or the advanced DNA, which is present in our cells in the nucleus, which are basically arranged in the form of the chromosomes. Right? Now, see, since it has the DNA, it is quite obvious that it will also have the ribosomes, because ribosomes are very much important for the organelle which will have the DNA. As without ribosomes, the DNA is useless because it cannot produce the proteins on its own without the ribosomes. Right? Now, that being said, the one unit that you see right here in front of the ribosomes is the 70S. See, ribosomes basically are of two types, 80S and 70S. S is nothing special but the sedimentation coefficient, sedimentation coefficient, which basically is used to determine the weight of a cell organelle. Okay, during the centrifugation. Centrifugation is the process which basically helps in determining the weight of different particles with the use of the centrifugal force. Okay, which you must have studied about in the physics classes. If not, ask a physics teacher. All right. Now see, ribosomes are of two types, the 70S and the 80S. 70S are present with the DNA, which is of primitive type, and 80S are present in the cells which have the advanced DNA. For example, the bacteria, it is the primitive, it has the primitive DNA. It is a prokaryotic cell, so it will have the 70S ribosomes. Similarly, mitochondria has the primitive DNA, so it will have the 70S ribosomes, which are smaller in size. Whereas in the eukaryotic cells, the ribosomes will be bigger, they'll be 80S. Why? Because we have the advanced DNA, right? Now, that being said, we're just left with discussing one thing, the matrix. Now, see, matrix is very important. Why? Because it is just like the cytoplasm. Matrix is the fluid, it is the colloidal fluid, or it is the thick fluid inside which everything floats in the mitochondria. Okay, colloidal fluid in which mitochondrial parts, that means the DNA or ribosomes, etc., etc., float, right? That is all about the mitochondria that you need to know. Next we have is the cell wall. Now you must have wondered that, ma'am, we studied about the cell membrane, we studied everything. Why are we studying cell wall right now? I'll tell you why. Because cell wall is just as important as rest of the organelles. Okay. Also, cell wall is present in the plant cells and most of the bacteria and a few microorganisms, but not all. Okay. Now see, starting with the cell wall can be composed of multiple components depending upon the organism in which it is present. Okay, since we are talking about the plants right now, we'll be talking about what are the components which make up the cell wall of a particular plant, right? So cell wall is surrounding the cells. Okay, and it is very rigid, it is very tough, and only in few cases it is flexible. It is flexible in the case of the euglena. Euglena is a kind of microorganisms. Okay, microbe, it's a microbe, right? And it provides the cell with the structural support, which is very important, and the protection, which is secondary important. And it also acts the filtering mechanism. Filtering mechanism, how? See, the thing is, cell is fully permeable. Okay, oopsies. So cell is fully permeable. Cell wall is fully permeable. It is not going to let's say stop the entry or exit of the molecules in any case, but it can certainly stop the entry and exit of the molecules which are larger than the pores present in it. Okay, cell wall will also have certain pores inside it, and if much of the molecules, let's say the bacteria or the viruses, if those larger molecules try to enter the cell, it will act as the filtering mechanism and it won't let them pass. Okay, now see, what you see right here is a cell. You can clearly see the whitish portion, which is the cell wall, and you can see the chloroplast or the plastid inside it, which we'll be discussing later on, okay, in this particular class itself. Now see, starting with the cell wall components first of all, and then we'll discuss about how it is made and stuff. So we have the plant. Plants have the cell wall made up of cellulose and hemicellulose, right? What are these? These are the long chains of a macromolecule which is called as the carbohydrate. Okay. Next thing that we have is the cell wall of a fungi, let's say. Okay. As I did tell you that the cell wall components could differ completely from organism to organism. Just to prove that point, I'm giving you two examples, one of the plants, one of the fungi. Okay. We have chitin, right? Chitin is also a kind of a macromolecule. Now, that being said, it is very important for you to understand the structure of the cell wall. Cell wall will have a middle lamella, primary cell wall, and a secondary cell wall. In few cases, there could be the tertiary cell wall as well. Okay, but the tertiary cell wall is not as important. So we'll just move ahead with the primary, secondary cell wall, right? Now, it's very important for you to keep in mind that cell wall is very, very important for the sake of protection of the cell, but it also limits the movement of the cell. That is why plants have it, because they cannot move from one place to the another, and animals do not have the cell wall because they can move from one place to another, right? And now we'll be moving forward with the structure of the cell wall, right? So the diagram that I'm about to give you is very important. There we go. Okay. I hope all of you can see this diagram very, very clearly. Okay, you see, this is a plant cell and this is the cell wall structure. Okay, now in the cell wall structure, what you see right here, we have the primary cell wall, we have the secondary cell wall, and then we have the plasma membrane, right? See, primary cell wall is something which basically is formed, let's say, which basically is formed in the very beginning. Okay, and before, see, primary cell wall could be made up of the cellulose, it could be made up of this hemicellulose, etc, etc. The primary cell wall is very, very thin since it is formed in the beginning, and it is elastic as well in order to let the cell grow a bit more in the size, because it just happens in the beginning. Okay, in the very starting of the cell formation, right? So during the development of the primary cell wall over the time, certain openings are left in the places in between the adjacent cells, which help in the continuity of their cytoplasm. Remember when I did tell you about the plasmodesmata, the cytoplasmic connections within the cell? That is the space which is left. Okay, so I'm just going to write here for you people: primary cell wall. This primary cell wall, it basically is thin, elastic, made up of cellulose, hemicellulose. Okay, and it allows the cell to expand and grow in size. Okay, to cell to grow in size. Apart from that, we have the secondary cell wall. Secondary cell wall, my dear little kids, is formed later on. Secondary cell wall, let's say, is situated towards the inner side of the primary cell wall, towards the cell, right? It is very, very thick, and it is also made up of cellulose and hemicellulose, but it consists of multiple layers of hemicellulose and cellulose. Okay, and the secondary cell wall starts being deposited onto the inner ends of the primary cell wall towards the cell after the primary cell wall is completely formed. Okay, so the secondary cell wall is non-elastic, it is very, very thick, and it is formed only after the completion of the primary cell wall. Okay, thick, inelastic, formed on the inside of the cell after primary cell wall is completely formed. Completely formed, right? Now listen to this. We have the primary cell wall, this one right here, okay? The red line that I'm drawing in this particular diagram is the primary cell wall, okay? As you can see, it is present on the outside of the cell wall. This is the primary cell wall again, okay? This is the adjacent, these are the two adjacent cells which are present, and in between the two adjacent cells we also have the middle lamella. Middle lamella is something which is formed whenever the two cells are trying to be formed, whenever the two cells are newly formed or newly synthesized, okay? That middle lamella is the first thing which divides the cell into two different parts, okay, whenever they are being synthesized. And then after middle lamella, the first thing which is laid out is the primary cell wall, which you see right here, primary cell wall. And after primary cell wall, we have the secondary cell wall, which is laid onto the inner side, the blue colored lining that you see here. You can locate that into the cell diagram, which is also shown, okay? So this is the secondary cell wall. Now, these are the different type of cell walls. These are the functions that we discussed about. You can also see this in this particular diagram which is shown. And the next thing that we are going to discuss are the plastids. Again, plastids are the semi-autonomous organelles. And when I say these are semi-autonomous organelles, that means they are also going to have a DNA just like the mitochondria. And that is going to help them in the controlling of their own self or their own function just a little bit, okay? And if they have that particular DNA, it is quite obvious that they'll also have the ribosomes, okay? And what kind of ribosomes? They'll basically have the 70S ribosomes. First of all, double membrane bound organelles, just like nucleus and mitochondria, okay? Second of all, found in the plants and algae, as I did tell you, also in bacteria, okay? Do not forget to mention that. Apart from that, responsible for manufacturing and storing of the food, and that is why called as the kitchen of the cell. Kitchen of cell, right? And next, they have the pigments which are used in the photosynthesis, which we call as the chlorophyll. And what does the chlorophyll do? Chlorophyll helps in the capturing of sunlight, which basically helps in doing the photosynthetic process. Next thing, and there could be multiple different kind of pigments as well, okay? So we'll be discussing the plastid structure right here, and afterwards I'll be giving you different type of plastids which are present, which are important for you to learn and revise, okay? Now that being said, let's just dive into this. We have the outer membrane just like the mitochondria, we have the inner membrane just like the mitochondria, and we have the intermembrane space, okay? Now listen to this. The chloroplasts are also responsible for the production of energy, but that energy is not utilized by any other cell organelle. Rather, that energy is completely utilized and used into the fixing of carbon dioxide into the carbohydrates, which later on are used by the cell as the food. Okay, so keep that in mind. Next we have is the stroma. Stroma or stoma, one and the same thing. This is also the aqueous fluid or the colloidal fluid, just like the matrix which was present in the mitochondria. If you're learning something, always try to learn things by comparing them with the other similar things which are present around you, okay? In that manner you'll be able to learn them quickly and retain them for a longer period of time. Now see, next thing that we have, the important things that you're seeing right here is the granum and the thylakoid. Thylakoid is a coin-like structure which is present like this, and inside the thylakoid we have a lot of chlorophyll. Now this thylakoid individually is one unit which help in the capturing of sunlight and the production of food, okay? But these thylakoids are arranged in the stack like the coins, okay? Like this. So one single unit is called as the thylakoid, and the stack of thylakoids is called as the grana or granum. Okay, grana right here is the plural and granum is the singular. Whenever you're trying to learn the scientific terms, just render their meaning first. So it is the plural and this is singular. Okay, now see, these are also going to have the lamella. Lamella are nothing special, but they are the connections which are present between the different thylakoids and the granum, which help in the maintenance of the continuity of function in a very systemic manner, in a very synchronized, or let's say in a very stable manner. Okay, so these thylakoid and granum can communicate with each other with the help of the lamella, right? And inside of the thylakoid is called as the lumen, where every single reaction related to the photosynthesis occurs. Okay, now this will also have a circular DNA, and it will also have the 70s ribosomes. Right, do not forget to mention this, because this is a semi-autonomous organelle and it is very, very important. Okay, so keep this in mind. Apart from that, I'm also giving you the different type of plastids which are extremely important for you to learn, okay, for the sake of your Olympiads and your exams in the higher classes though. Okay, so you see, we have the plastid. Plastid is the organelle which is present only inside the plant cells, only inside the plant cell, and sorry, the plant cell, and chloroplast is one of the type of the plastid that we just studied. The function is photosynthesis and the lipid synthesis. Okay, then we have the etioplast. Etioplast is the crystalline prolamellar body, which is the precursor of chlorophyll. Basic meaning of this particular thing is that etioplast leads to the formation of chloroplast. Etioplast is the primary structure which then develops later into the chloroplast. Then chromoplast is the one which helps in the pigment synthesis and storage in order to give the color to the different flowers and fruits, okay, and vegetables. Leucoplast, on the other hand, it helps in the storage and synthesis of the fatty acids and amino acids, again lipids. And these amino acids, they lead to the formation of multiple proteins. Okay, amyloplast, starch and sugar, carbohydrates basically, elaioplast, lipids and oils, proteinoplast, proteins. Okay, and next thing that we have is the questions. I mean, can you imagine? It was a long session, right? So the question that I have for you today is, what is a semi-autonomous organelle? Okay, or what is autophagic vacuole? Okay, and just one more question that I have for you is that what structure in mitochondria synthesizes ATP? Okay, you know the answers to all. Semi-autonomous organelle, self-controlling organelle, partially self-controlling organelle, mitochondria and chloroplast. Always mention the examples whenever you're asked a question like this. The mentioning of examples will ensure your marks and the bonus points. And what is an autophagic vacuole? Autophagic vacuole, auto self, phagic, digesting or engulfing. That means the suicidal bags, the lysosomes. And what structure is in the mitochondria synthesized ATP? ATP synthase, or the peroxisomes or the oxysomes. Okay, if you do not remember the peroxisome and oxysomes, you can always mention the ATP synthase. That will be the correct answer. And thank you so much for learning with us. I'll be meeting you in the next session. Till then, keep safe and enjoy, and keep on learning and revising. That's all for today. Bye bye.